Hello guys, today we are going to discuss factors affecting gene linkage on chromosomes. So first we are going to discuss two or three definitions related to this linked genes or the gene linkage or the uh, inheriting of the genes together to the next offspring. So here linked genes are the genes that are likely to be inherited together because they are physically close to one another on the same chromosome. Here for example, when you are going to consider the chromosomes are going to have a group of genes of its own. If you are going to consider, a chromosome can have one or two or more number of the genes as per the DNA amount is present within it. Now here, when you are going to consider the characters will be present on that chromosomes. For example, when you are going to see the picture here, in this picture you can see that genes are present on the chromosomes, which within the first one you can see two genes which are located near to each other, and in the next one the two genes are located on two different chromosomes which are not linked. Now here it is the two genes which are present far away on the same chromosome which are again not linked. So here the linkage is one, how closely the genes are located on the same chromosome. So here when you are going to consider, if the genes are alternatively located, for example here there are gene 1, gene 2 and there is gene 3. So here 1, 2 to above to the gene 1, if there is gene 3, so here the difference between the gene 2 and gene 3 will be of the gene 1. So the linkage or the linkage between the gene 3 and the gene 2 is going to differ by one of the gene or the whatever the DNA amount or the distance of the chromosome has been found. So this is how the genes are linked with each other. So here if the genes are going to get inherited, then it has to be located closely to each other on the chromosome. So here this is what, here if they are not closely located each other, if they are found like this apart, the chances of the crossing over during the packaging stage of the prophase 1 is high. So the recombination is going to happen. It means that the whatever the parental other chromosome will be present, it is also going to have the gene 1 and gene 2 of the same characters. So here it is going to take part in the crossing over, that is the exchange of the genetic material between the two parental chromosomes in their tetrad form. So the sister chromatids are going to take part in the crossing over between the two parental chromosomes and the genetic material gets swapped. So because of that, the new recombination will happen. So this is how the gene linkage is going to weaken in the strength. So next, coming to the one more definition, that is genetic linkage is the tendency of DNA sequences that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. Now here in the previous slide we saw that these are the physically located loci. Here it is the definition speaking about the tendency of the DNA sequences. So in the next one, here it is again the number of the bases between two genes in a linear DNA sequence. So here a gene is one which is, if it is present on the chromosome, can be mentioned to be as a loci, that is the located position. In this definition, as per that, here it can be a expressive DNA sequence that are close together on a chromosome to be inherited, and here depends upon the number of the bases between two genes in a linear DNA sequences. Now here when they are explaining that between the two genes, DNA sequences. So the whole chromosome is going to be of the DNA sequence itself. But if the genes are mentioned with the certainly like this kind of the explanation, two genes differed by a DNA sequence is nothing but the two genetic markers which are identifiable because of their certain expressive character. So here all the three definitions are explaining the same concept of the linkage and not linkage of the genes. So here let it be the gene loci here the what called expressive form of the DNA sequences, that is the gene which is going to be inherited. Here if you are going to consider it to be as the genetic marker for the DNA sequences, then it is going to be as the same. So here all the three definitions are mentioning it to be as the genes located together are going to be inherited likely to the next generation. If the genes are apart on the chromosome, are not going to inherited likely to the next generation. Now here factors affecting gene linkage on chromosomes. Certain factors affecting the genetic linkage or the gene linkage, either they will be helpful to retain the parental combinations or they will disturb the parental combinations to give rise to a recombination through crossing over. Here parental combinations can be of homozygous or heterozygous form. So here to study a inheritance and to understand the crossing over has been happened or not, we have to take two different parents who are having a completely dominant character and a completely recessive character, so that the further generation and the further test one or generation can also be studied whether the how much of the ratio of the parental combination has been inherited and how much of the recombination has been inherited and the how much ratio of the parental recessive form of the inheritance has been taken place. So here you can understand in the pea plant we have many studied the parental combination, recombination and also the recessive parental combination. So here we see both the dominant and recessive homozygous and the heterozygous form of the recombinant one. But that is totally we are understanding through the qualitative form of the inheritance of the genes. But certainly when we are going to discuss about the linkage, here we are totally discussing the certain other factors which are other than what we have understood during the Mendelian inheritance. Here there are few factors, that is distance factors, centromere factor, age effect, temperature effect, X-rays effect, chromosome effect. These are going to leave some effect on the crossing over within the packaging stage of the prophase one. So we are going to discuss this one by one. In distance factor or the effect, if you are going to consider chromosomes are the physical bodies which are going to bear the gene loci for the characters. Now when you are going to see the chromosomes are going to have two arms with a centromeric position. One will be the P arm and other will be the Q arm. This will be the centromere. So here when we are going to see this single chromosome within the meiotic stage, they are going to have the sister chromatids, that is the duplicated chromosome. If the organism is diploid, let we consider the diploid organism only to take a better note and understanding of the concept. So there will be one more parental chromosome also, so that is also going to develop or duplicate its parental chromosome and it is going to form the tetrad form or the sister chromatids it is also going to have. So here this is how the chromosome is going to appear. Here you can see the chromosome has been stained, so it is showing the both euchromatic and heterochromatic regions within it. So whatever the darkest regions you are seeing, that is the heterochromatic region, and whatever the lighter regions you are seeing, those are the euchromatic regions. So this picture is depicting as a symbolic one. The actual pictures of the chromosomes will have more different appearances. So it is a diagrammatic one to make things very simple and understandable. Now here in this picture you can see the depiction. Here the chromosomes and the genes has been arranged. Whatever the bands has been shown here, those are the genes. This is the parental chromosome as we are referred here, and this will be the sister chromatid. So here the genes are arranged linearly in each arm, that is here, 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 in different colors. So in sister chromatids also, when the parental chromosome duplicates, the sister chromatid also going to have the same genes, similar genes duplicated on the both the arms P and Q. Now here these genes will be placed linearly on the chromosome, one after the other. So here we are considering it to be as a linear because we are going to denote it, identify it with some identifications. So why we are calling it to be as a linear? Because its chromosome is a condensed form of the slender with some unique regular structure, which is going to allow the DNA sequences to get condensed in such a form that the genes are going to arranged linearly on it, one after the other. So that is why these gene loci are linearly placed on the chromosomes. So further, in this distance factor, now considering the distance of linearly arranged genes, the first gene loci will have distance as the next or one after the other gene loci on chromosome. It means if you are going to consider the chromosome, as we have discussed that the gene loci are linearly arranged, that is one after the other. So if you are going to denote or symbolize them with alphabets A, B, C, D, E, F, as the bands are appearing here, as corresponding to that, if you are going to mention that, so the linear genes will be A, B, C, D, E, F. Now in the sister chromatid also, the loci are going to fall the same. So they are arranged one after the other. Now in the next slide, the genes are closer to each other, the linkage will be stronger, and the crossing over effect will be less on the closely linked genes. Now here, if you are going to consider A and B, these are the two genes located on the P arm. C, D, E, F is located on the Q arm. So when you are going to consider A and B are closely located to each other, C, D, E, F are located on the other arm, they are not closely located to the A and B. And also C and D are located to each other, which are very closely located. D and E is located to closely located to with each other, and E and F is closely located to each other. When compared to C and F, D and E is going to form the distance, that is the DNA sequence. The second and the third definitions what we have discussed, that is applicable here, that is D and E is going to form the DNA sequences. They are the genetic markers to express themselves, but for the linkage strength between the C and F, they are going to be a distant. So there is a chance of the crossing over or the exchange of the F gene with the other parental chromosome. That is the other form of the allelic form of the parental chromosome take exchange here. So as the C is far from the F and the D is far from the F, there is a chance of the exchange of the parental DNA or the chromosome between the both the sister chromatids of the different parental chromosomes. So here the distance between the A and F is also the farthest one, and also it has been separated from the centromere. So here these factors of the distance, whatever the far away distance genes, they are likely to get recombined, very likely to get recombined. Whatever the genes which are closely located, they are likely to get inherited. It means the ratio of the inheritance of the closely linked genes are high when compared to the ratio of the farly distantly placed genes. Inheritance is going to be very less. In the other words, the genes C, E and F are likely to going to inherit at the high ratio compared to the genes C and F. That is, their ratio of inheritance is lesser than the ratio of the E and F. So this is what the linkage describes. When you're going to see here in this, you can observe very clearly in this diagram. You can see E and F here, and this is one parental tetrad form, this is the other parental tetrad form, and this is the linear genes present here, and crossing over takes place here. So E and F is present in these arms. So one of the chromosome out of the tetrad form is going to take part in the crossing over. There can be the chance of the both the arms taking part, but major ratio is of the one arm is going to take part. So here this E and F is going to get exchanged. So here the whatever the linearity we have observed, A, B, C, D, E, F, here in the parental chromosome C and C, D, E, F all are linked. When the recombination takes place, the linkage between the C and E, F is going to be very weak compared to the C and D and E and F. It means they are close to each other, so they got inherited together, and here they are close to each other, they are on the parental chromosome itself, and they are going to get inherited together. So when you compare C and F, they are far from each other. D and F, they are far from each other. C and E, they are far from each other, and D and E is having the distant DNA sequence present between them. Because of that, they have got inherited here. Now here all the three definitions are very well applicable here. Whatever the three definitions we have discussed earlier in these slides, it is very well applicable. So here all the three has been described. Coming to the next slide, if the genes are located far away from each other, the linkage is weak, then the crossing over effect will be more. The same thing what we have discussed in the previous slide. Here, as we have spoken, as the C and F is far away, and because of that, the crossing over, during the crossing over, the C has been left in the parental chromosome itself, and F has been taken into the exchange or recombination. So the new genetic recombination has been taken place. So because of that, the crossing over effect will be more between the far away located genes. This is what the explanation. So as the distance increases between the genes, the strength of the linkage decreases, and as the decrease in the distance between the genes, so the strength of the linkage increases. So This is what the simple form, whatever we have discussed. I hope I was audible on this particular point earlier. Centromere effect. These effects are due to the chromosomal rearrangements. In this, the centromere position will be changed by exhibiting different linkage group on chromosome. In a section of chromosome contain 2 or more genes are placed close to the centromere, the chances of crossing over are reduced. Now here, for example, you can observe we have taken the same picture for the understanding of this same centromeric effect. Now here, if you are going to consider this A, B, C, D, E, F, in this centromere is present here. These are the 2 parental chromosomes in their tetrad form. So here the nearby genes to the centromere is B and C. So here, when you are going to consider the crossing over at this location is very rare. The centromere will not take part in the any kind of the crossing over because it is highly condensed part of the chromosome, and whatever the genes which will be located here and whatever the DNA or genetic material which is let it be a genetic marker or expressible gene loci or let it be a heterochromatic DNA sequence, the exchange of this will not takes place. Now here, if you are going to consider in this diagram, the chances of the longest arms during the crossing over are more, and even this sometimes the shortest arms also going to take part in the crossing over, but the centromere will never take part in the crossing over because they will be tied up the proteins and they are not detachable during the packaging stage. Now here these are going to whatever the genes which are going to be present near to this centromeres, they are not going to take part in the inheritance. So the linkage of the genes which are present near to the centromere are high, and the parental combinations of these genes, linked genes nearby the centromere, are going to be inherited as it is. So here this is what the centromeric effect. Certainly in some rare conditions, if there is any aberration during aberration in the chromosomes, let it be duplications, let it be deletions, let it be any kind of the inversions, translocations, they are going to cause the different kind of the homologous pairing. Here you can understand what is homologous pairing. During the meiosis cycles, the parental chromosomes, while they are going to form the tetrad formation after the duplication of the DNA in the prophase 1 of meiosis, the parental chromosomes are going to come near to each other to pair to take part in the crossing over process during packaging. During this time, the different formation of the homologous pairing takes place. The homologous chromosomes pairing takes place. So here, during this stage, the different shapes of the chromosomes can be observed. Different homologous pairing shapes can be observed. Those are going to form depending upon the genes arranged within the chromosomes. So here, as we have spoken already, these effects are due to the chromosomal rearrangements. So here, certain times the chromosomal rearrangements will take part very differently depending upon the presence of the genes, that is the linearity of the genes, and also certain different physiological effects are going to lead to the certain different form of the homologous pairing formations. So depending upon that also, the gene recombination is going to be affected, or the crossing over is going to be affected. Because of that, the inheritance of the linked genes is also going to be affected. But in the centromeric effect, it's the ultimate understanding or the complete understanding that there is no recombinations observed, or very depleted ratio of the recombinations has been observed for the genes which are very closely located with the centromeres. So the parental combinations will retain. Now, age effect. Age effect is in the sense of the physiological changes happens in the meiosis cell division, that is during the chromosomal crossing over. It holds or disrupts the linkage strength of closely located genes and far apart located genes on the chromosomes. As observed in the organisms of lesser age, during cell division, the process of crossing over frequency of the chromosomes are high compared to the older age of the organisms, where crossing over frequencies reduces. The understanding is that at the lesser or young age, the frequency of chromosome crossing over is high, the linkage strength reduces, and with the increase in the age of the linkage, strength reduces gradually and the linkage between genes increases. Now here, if you consider the age effect, during the young age, so that is maturation of an individual taking part of the gametogenesis, and during that time the frequency of the crossing over within the individual during the phase 1 packaging stage will be high compared to the age which is in the older. So here, when we are going to consider in the older ages, during the higher age, the organisms cell divisions, the process of crossing over frequencies of the chromosomes are very less compared to the lesser age, because with the older age the physiological conditions of the organ and also the cytological physiology, the cell physiology, are not going to be at the optimum. So because of this, there will be the reduction in the cell division process also, and also the frequency of the recombination is also going to reduce with the corresponding cell divisions. So here in the young age, the factors affecting of the age will be very less, that is the young age will be having the effect as the young age factor, where the recombination frequencies will be high, leading to the linkage strength to be less, whereas the at the higher age the recombination frequency will be less, where leading to the linkage strength to be high, where the parental recombinations can be inherited to the next generation. So here this is regarding the age factor. In this slide we will discuss the temperature factor or the effect affecting the gene linkage on chromosomes. So it's an external factor, it is a completely external factor, has effect on the gene linkage of the chromosomes. So here this external factor is again a multifactor, which is a quantitative kind of the factor, which is going to have some influence on the gene linkage on chromosomes, that is within the gametogenesis process, that is during the meiosis cycle, which is going to give rise to the gametes. Now here, at minimum required temperatures, the crossing over frequencies will be normal and the linkage among the genes in a chromosome will be disturbed. The new recombinations will form. So here, at minimum required temperature, it means that it's a threshold temperature required to a particular physiological functions to run on a particular rate, which is the threshold rate of output will be there. So it is the one of the particular threshold temperature which is required to gametogenesis. So here this required temperature or the threshold temperature is going to differ with each and every organism. So at this threshold temperatures, the crossing overs within the cell cycle packaging stage of phase 1 of meiosis cell cycle is going to be very high. So here, due to this, there will be the disturbance or the linkage or the linearity of the genes on the chromosome is going to be disturbed because of the new recombinations are going to be inherited. So with the increase of the temperatures, the crossing over frequency will reduces. The gene linkage in the chromosome will remain intact and exhibit parental combinations in next generation. In the sense, if we are going to consider an organism is going to be in a higher temperature environment, due to that there will be the effect on the physiological function of that particular organism's organ which is responsible for the gametogenesis, like the gonads. Now here the organism's organ is going to take part in the gametogenesis, but due to the difference in the physiological function because of the higher temperatures, there will be the less frequency of recombinations compared to the whatever the threshold minimum temperature we have already spoken about. Now here, due to this less frequency is going to less frequency, it means it is going to reduce the recombination. So here, as per the minimum required temperature and as per the increased temperatures, in minimum required temperatures, the linkage or the linearity of the genes will get disturbed from the parental chromosome and get exchanged between the two parental chromosomes and get inherited into the next generations. But with the increase in the temperatures, there will not be any disturbance, or the linearity of the genes on the chromosomes will not going to get into the exchanging of the genes between the two sister chromatids of the two different parental chromosomes, so that it doesn't going to inherit the new recombination. So the parental combinations are going to be intact. The conclusion is that each and every organism is going to have a threshold temperature, but it doesn't mean that each and every organism is going to react or give the same results as it has been discussed in this slide, because multi-factor inheritance or the multi-factor effects are not the actual effect which are going to give the same results every time. So here we are accepting the concept and not rejecting the concept, but we are going to place the concept within the multi-factor inheritance kind of the understanding. Next slide, X-ray factor. X-ray has effect on the physical factor which has the ionizing feature. Ionizing feature in the sense the compound will be broken into their ionic forms. So the component will be separated and further their actions will be studied. The X-rays has the effect on the organisms. Always remember, each and every organism is going to have the effect of the X-ray depending upon the frequency or the time period it is going to be subjected to. Due to ionization process, we observe the new combinations in next generation, disturbing the parental combination. So here it is totally a inducing kind of the factor. Due to disturbing the parental combinations, the strength of the gene linkage reduces on the chromosomes. Chromosomal effect. During the process of condensation, a unique structure of the chromosome will form of its own. If the change in the process of condensation of chromosome structure, the genetic arrangement will also change. Such changes will affect the process of crossing over by favoring the parental linkage of genes. Now here, it is a very rarely happening phenomena, because in labs they have done some experiments and have got the results. So here, when you're going to consider linear arrangement of the genes on the chromosome, and they are going to take part in the crossing over during the packaging stage, where the packaging stage of the chromosome are going to be in the fusiform. In simple words, if you say that the DNA will be in its scaffold model, but they are freely substituted on the histone particles. They are not completely packed as the chromosome. They are readily available in the form of the DNA sequences, but on the protein histones. So here, in simple words, if you say, it is a zip-like formation or the appearance when you are going to observe under the microscope. That condition of the appearance will be formed from the first stage of the prophase, leptotin stage. From that stage, the certain proteins and enzymes are going to take part for this condensation, like cohesin and condesin and the topoisomerase, which are going to describe the topology of the chromosome. Topology in the sense the physical appearance of the chromosomes. In certain experiments, they have made the substitute of this cohesin and the condesin. It means that these are the responsible one to give the condensation in the proper way, but after subjecting the chromosomes to some experimental understanding, they have tried condensation of the chromosomes or the DNA material without the cohesin and the condensin proteins. They are all, so these proteins or the enzymes are responsible to give the gradual progress in the condensation of the chromosomes. If there is the disruption within this particular structural formation, there will be the disruption in the linearity formation within the chromosomes. Now here, if you are going to consider out of the two parental chromosomes of a diploid organisms, both has to form the sister chromatid. One is at its regular form and the other is not at its regular form because of the disruption of this particular proteins. So what will happen? There can be the difference within the arrangement of the structure of the chromosome within the packaging stage. So because of that, there can be the chances of the difference in the crossing over. So there can be the chances of the even the linked genes can take part in the crossing over. That is what they are trying to describe, just to make the understanding. Chromosomal effect in this form is very negligible compared to the other form of the effects. So such changes will affect the process of crossing over by favoring the parental genes. It means when the homologous chromosomes does not get into the pairing, there can be chances of no crossing over, and the parental combination can get inherited. This also can happen, but usually within the cell cycle, especially the condensation process, is highly efficient. But as per the experiment, for the understanding, they have been given this particular explanation. Further is the explanation of the factors affecting the linkage envisages that an organism subjected to factors will show either the favorable for the crossing over phenomenon or unfavorable for the linkage group on the chromosome and vice versa.